I hope that you are all as excited as I am for this next session. Joining us are four of my all-time favorite people in the celiac disease community. They are all incredible physicians and researchers who have dedicated their careers to bettering the lives of patients with celiac disease. I could spend hours telling you all millions of reasons why each one of them is amazing, but I'm going to stop talking and let them talk to you about their work to develop medications to treat celiac disease. Sandy is going to start us off by doing the honors of introducing each of the speakers.
Well, I'm indeed honored to be able to do this. And each of our speakers has such a long list of extraordinary qualifications and expertises and experience that here we go. Our first speaker is Dr. Ciaran Kelly, Professor of Medicine at Harvard Medical School and Director of Gastroenterology Training at Beth Israel Deaconess Medical Center in Boston, Massachusetts. Dr. Kelly has engaged in patient care and research in celiac disease for more than 20 years. In 2004, he was a founder of the Celiac Center at BIDMC and continues to serve as its medical director. In 2013, he co-founded the Celiac Research Program at Harvard Medical School, which brings together celiac disease researchers and educators from Harvard University, teaching hospitals, including Boston Children's Hospital, Massachusetts General Hospital, and BIDMC. Dr. Kelly is an internationally recognized expert in the diagnosis and management of celiac disease, and in his clinical practice specializes in difficult to treat enteropathies. He also leads research programs in the pathogenesis of celiac disease, its diagnosis, and new approaches to treatment. He is currently serving as a president of the Society for the Study of Celiac Disease. Our second speaker is Dr. Daniel Leffler. Dr. Leffler currently serves in dual roles as the medical director of the celiac disease program at Takeda Pharmaceuticals and the director of research at the Celiac Center at Beth Israel Deaconess Medical Center. Dr. Leffler currently divides his time between patient care, quality improvement initiatives, and clinical and translational research in celiac disease and other digestive diseases. Dr. Francisco Leon is our third speaker. Dr. Leon is the Chief Scientific Officer and co-founder of Provention Bio, Inc., an immunologist by training. Dr. Leon has a breadth of experience and expertise from his academic at NIH and industry careers in the fields of immunology and immune-mediated disease clinical research. Previously, Dr. Leon was a co-founder, chief executive officer, and chief medical officer of Cell Immune LLC, a clinical stage immunotherapy company dedicated to developing transformational therapies for celiac disease, intestinal lymphoma, and other serious immune-mediated diseases. Prior to Cell Immune, he was vice president and head of translational medicine at Janssen Immunology, which is a division of Johnson & Johnson. Last but not least, our Dr. Jocelyn Sylvester is the director of research for Boston Children's Hospital Celiac Disease Center. She completed her Ph.D. in biology at the University of Cambridge in Cambridge, England, prior to studying medicine at Dalhousie University in Halifax, Nova Scotia, Canada. She's our Canadian. She completed her residency at the University of Manitoba in Winnipeg, Canada, and her fellowship in gastroenterology, hepatology, and nutrition at Boston Children's Hospital. Dr. Sylvester continues to work as a general pediatrician in remote areas of northern Canada, as well as at Boston Children's Hospital. Her research interests include the diagnosis and management of celiac disease. She holds a K Award from the NIH and has done extensive research on gluten immunogenic peptides and the transfer of gluten in urine and stool. What a star cast. Thank you so much for being here. We're so thrilled to have you.
All right, so Dr. Kelly, we're going to turn it over to you.
Great, I'll share my screen and see if I can get my slides up. Good. Good morning, everyone. Great to be able to speak to you this morning, and I'm honored to share the podium with such a star star-studded cast of celiacologists. I'm going to talk to you about treatments to induce tolerance to gluten. Those of us who are familiar with celiac disease know that the problem with celiac disease is that the body's immune system foolishly attacks gluten and is intolerant of gluten. And so I'm going to talk to you about one of actually several approaches to undo that ill and induce tolerance rather than intolerance to gluten. I'm going to talk to you about one approach, but I'm happy to say that there are multiple groups around the country and around the world who are trying complementary approaches to achieve the same aim. I'm also happy to say that I work with several pharmaceutical companies in trying to develop new treatments for celiac disease. This list would have been much, much shorter if I was to give this talk 5 or 10 years ago. It's, in my mind, a happy outcome that there are more and more companies in pharma who are now interested in helping us to develop new non-dietary treatments for celiac disease. There are perhaps a couple or 3 reasons why pharma is interested in celiac disease. One is it's a very common condition. We now know that approximately 1% of the population of the world have celiac disease. Many of those are undiagnosed, but nonetheless, they suffer from the condition. We also are happily aware that we actually know a lot about how celiac disease develops. An individual with celiac disease ingests gluten in their diet. Gluten is difficult to digest, and so it remains intact in the lumen or channel of the intestine. Small parts of gluten cross over and activate an immune response in the body through what are called antigen-presenting cells. And these antigen-presenting cells activate what are called T cells in the body. And these T cells cause mischief because they react to gluten as if it were an invading virus, basically. And a flu-like condition akin to rotavirus infection develops. Many of these steps are well known in celiac disease, and because of that, they can be attacked or addressed pharmaceutically. So one approach to this is to package gluten in a pocket of what is called PLGA. So PLGA is the material that's used to make absorbable sutures. So it's inert and absorbed. And if you package gluten within this, you can deliver gluten to places of the immune system where it can induce tolerance rather than inducing an immune reaction. So the idea of this approach is to package gluten in these biodegradable nanoparticles. They are then taken up by the antigen-presenting cells that I mentioned earlier in the spleen and in the liver. But most importantly, those are sites where so-called Marco scavenger receptors reside. And these are special cells that carry this Marco receptor, which identify them as the sort of dustbin keepers of the body. And in these sites, in the spleen and the liver, dead cells will be taken up and disposed of. As the body is disposing of these dead cells and particles, it does not want to treat these dead self cells as harmful. It wants to ignore whatever immune signals these cells might send out, because these are simply dead self cells. And so these Marco-bearing cells are programmed to reduce, downgrade, avoid an immune response to these dead cells. So by presenting gliadin to this population of cells, gliadin becomes an antigen, a substance that should be ignored. that an immune reaction to should be downgraded or avoided. And so by engaging these Marco cells, these Marco positive antigen presenting cells, the goal is that one can suppress gliadin activated T cells by removing those cells or by turning them off. So in a way, this is a trick to trick the immune system into reconsidering its response to gliadin, not as an aggressive particle, but as something that should be ignored and tolerated. So the study that I'm going to talk about using this approach, we enrolled patients with well-controlled celiac disease on a gluten-free diet for a period of time. Those individuals were given, Those individuals were given 2 doses of the TAC 101, that's the immune modifying particles that I talked about, were given 2 doses of the TAC 101 1 week apart. And following this, they were exposed to a gluten challenge. A fairly high dose of gluten was given initially, and then a lower dose over the following few days. And before and after the gluten challenge, we looked at gliadin reactive T cells, and we also looked at histology or biopsies, looking at villous atrophy in the small intestine. So it was a small study. This is a early phase pilot study. So there were just 34 participants divided randomly between those who received the active timgliadin and those who received placebo. Placebo being, an inactive mimic of the active ingredient. They were mainly in their 40s. They were predominantly female, as is the case for most people diagnosed with celiac disease. The treatment was very well tolerated, and there were no real differences in side effects between active and placebo, with the possible exception of back pain. And this occurred as a symptom during the infusion of the nanoparticles and resolved quickly and did not require any intervention. So the main readout of this study were what are called ELI spots. ELI spots reflect gliadin responsive T cells in the circulation. And these are T cells, or cells of the immune system, that react against gliadin, release what are called cytokines, and give a cytokine storm, which ultimately results in symptoms and injury to the intestine. So we were looking at the activation of these gliadin responsive T cells in the circulation. And we looked at this at baseline, before any gluten was given, after the treatments, before any gluten was given, and then we looked at it again 6 days into the gluten challenge. So these are the main results of the study, and I'm just going to focus on the right-hand column. The left hand gives more detailed data. The right hand shows the change in the activation of these gliadin responsive T cells. And with placebo or sugar or dummy treatment, you can see there was an increase in gliadin responsive T cell activation, whereas with the TAC 101 treatment, that increase was markedly, significantly reduced. So this is actually the main outcome of the study, with the ability to prevent activation of these gliadin responsive T cells. A second measure that we looked at had to do with villous atrophy. Normal villi are shown here on the left, and on the right is the biopsy of somebody with celiac disease. And you can see that villous height to the depth of the crypts, which is where the villous cells come from, it normally is around 3. In celiac disease, it's much lower. So we compared villous height to crypt depth at baseline compared to after the exposure of 2 weeks of gluten. And I'm again just going to show, I'm just going to focus on the right side of the graph. And this shows a drop in villous height, which you'd expect with gluten exposure, which was markedly reduced in those who were treated with the TAC-101. Finally, and I'm not going to spend a lot of time on this, but basically this looks at the activation of these aggressive T cells that are empowered by gluten exposure and migrate to the gut. So these are looking at T cells that are headed towards the gut where they cause mischief. And you can see in the placebo, there's a marked increase in these gut homing T cells that's prevented by treatment with TAC-101. And these are looking at 3 different populations of gut homing T cells, each of which are prevented by treatment with TAC-101. So to summarize, what this study shows is that if you package gluten in inert envelopes and infuse it intravenously, those envelopes go to the spleen and the liver. There they engage these MARCO1 T cells, which induce tolerance. And instead of activating gliadin-specific T cells, they inactivate these cells, and as a result, there are fewer of these T cells in circulation. There are fewer of the gut homing aggressive T cells that are activated by the gluten, and as a result, there's less intestinal injury or damage. So this is a new approach to inducing tolerance to gluten and celiac disease. This particular study was successful in its primary goals and showed a reduction in activation of T cells and in deterioration of biopsy. It also showed reduction in gut homing T cells during the gluten challenge. So this is an exciting finding in my mind. Firstly, it's the first clinical study to show induction of immune tolerance essentially in any autoimmune disease, not just celiac disease. It shows the potential to tolerize patients who have gluten intolerance and celiac disease to gluten, and actually beyond celiac disease may act as a pioneer for antigen-directed tolerance in other autoimmune diseases that are less well understood. Thank you.
Thank you, Dr. Kelly. We are now going to bring in Dr. Leffler, who is going to expand a little bit on this, and he's going to actually start answering some of the questions that have been coming in during Dr. Kelly's talk.
All right, so hopefully everyone can see my slides. Thank you again. And the good news is that Ciaran very eloquently talked about some of the stuff I was going to talk about. So we will hopefully have a few extra minutes for questions. That I'll try to address some of them that have been coming in during this talk, and some we'll have afterwards. So let me just start with revisiting one of the things Ciaran talked about, is that we have a growing number of therapies in celiac disease. And this is for a few reasons, as was mentioned, partially because it is increasingly recognized that this is a large unmet need and that the gluten-free diet does not fix everything for all patients with celiac disease, but also because we have a great understanding of how celiac disease works. And that means we can actually develop therapies that are incredibly targeted. And almost as a rule in medicine, the more targeted your therapy is, the more effective it can be and the less side effects you are going to have. So if you have a therapy that just does something broadly because you don't really understand the disorder, you're sort of asking for some problems with side effects, off-target effects. But in celiac disease, we can be a lot more directed. And so I want to walk you through just quickly some of the main types of targets. And then I'll talk specifically about the 2 I'm working on, which is TAC-062 and TAC-101, some of which you just heard about. So starting with number 1, we can prevent gluten from interacting with your body. And there's a couple ways to do this. The major one that's been investigated so far, the most common is using a glutenase, an enzyme to break down gluten so it becomes harmless. But there are other technologies too. Things that affect the tight junctions that prevent gluten from getting in, things that bind gluten in the intestine to keep it from interacting, even increasingly some potential ways we might affect the microbiome to break down gluten so it becomes harmless. Number 2 is tissue transglutaminase. Now, we all know this as the blood test that we test for in celiac disease, but it is actually the enzyme that converts gluten into something harmful. And gluten in its native shape, and it's the way it starts in nature, is actually not harmful. It needs to be changed by this enzyme in your body, tissue transglutaminase, before it becomes active. And so if you block this enzyme, you could potentially block celiac disease from becoming active. Number 3 is where TACO 101, which you just heard about, TIMP-GLIA, sits. And this is turning off the specific immune reaction to gluten in celiac disease. So remember, no one is born with celiac disease. We develop celiac disease. We are born with the ability for the immune system to figure out what is harmful and what is safe, and what it should react against and what it's not. And so if we once had the ability to tolerate gluten, we should be able to flip that switch again if we have the tools to do it, and convince the body that gluten is again harmless and not make the body respond to it. And this is the realm of antigen-specific immunotherapies. There's a growing number of these out there across all autoimmune disorders, but TAC 101 is of these probably the furthest along. Then there's cytokine agents that block some of the key cytokines that cause inflammation in celiac disease, and I'm sure Francisca will talk about those. And then the final thing is things that may help the intestine repair. One of these could be GLP-2, but there's other ones that help the intestinal villi grow back, and so can maybe repair or speed the repair of some of the damage we see in celiac disease. And really, these are the 5 classes of celiac therapies to keep an eye on, and almost everything you'll see will fall into one of these 5 categories. So moving on to the ones that I'm working on, that I have sort of the honor of working on. The first one I want to talk about is TAC 062. This was previously known as Kuma 062. And this is an enzyme, a 3rd generation enzyme for treatment of celiac disease. So this is started out as a bacterial enzyme, and they use some very fancy computational techniques to make the enzyme incredibly efficient at breaking down gluten. And so when you look at enzymes, because there's been a lot of enzymes that have been studied in celiac disease, there are even some that are unproven and available over the counter, you really want to think about 3 things that define whether an enzyme could actually be useful in real life. And that is, does the enzyme actually survive in the stomach, right? The stomach is not an easy place. It's very acidic. There's lots of enzymes that break down proteins in the stomach. So can the enzyme actually survive that? Can it actually break down gluten peptides? So gluten is hard to break down. We can't break it down. Mammalian enzymes really don't do a good job of breaking down gluten. That's why it can cause this reaction. And the third is it specific? And the specificity is actually one of the newer things in enzyme science right now, because what you see with some of the older enzymes is you put gluten alone with these enzymes, they actually do a great job breaking down gluten. But if you mix them with other proteins, then suddenly these enzymes are breaking down all these other proteins and ignoring the gluten. It's almost like, now in the era of homeschooling, almost like having a focus task for your kids versus letting them do whatever they want and seeing what happens. And clearly they're going to get a lot more done if you focus the task. So Kuma, or TAC 062, is a very focused enzyme that really only breaks down gluten and ignores all the other proteins. And so because of this, we thought it was worth testing. And so what I'm going to talk to you a little bit about now is the results of a phase 1 trial and proof of mechanism trial for this. And so really this was a study in some celiac patients, but mostly healthy controls. And it was fairly simple. You gave people the enzyme, you gave people a smoothie, which contained gluten, which I'll show you in a minute, and then you took samples of the stomach at time points and saw how much of the gluten was broken down. And that was really the primary aim, is to see if this really looked as good in people as it did in the test tube. Also to look at some other things, the effect of some other medications like acid blockers on it, and evaluation of a couple different formulations, liquid and solid. So first of all, I just want to show you the test meal because it is important. Again, you want something that will actually work in a real-life meal. No one goes out and just eats gluten powder. They have a meal which gets contaminated accidentally with gluten. And so you really want to test whether or not an enzyme will work with a very complicated meal. And so for this, we did a smoothie that actually apparently tasted pretty good. Vanilla ice cream, egg whites, orange juice, whole wheat bread, lime juice, and vanilla extract. So a good mix of fat, protein, and carbohydrates, just as you would normally get in a meal. And note that it's a big dose of gluten that we used in most of these. 3 grams of gluten, almost 30 grams of bread is what that converted to. So really far beyond an accidental exposure. And I'll just jump to the sort of the top line data, I guess, is that as we had hoped, TAC-062 did achieve consistently really high levels of gluten degradation in the gastric space, up to 99% in most tests. And I'll walk you through this a little bit. So in the column on the left, you see the different groups. The middle column is the R5 ELISA test results, and the right column is the G12 ELISA results. So these two ELISAs you may see in food testing. They're commonly used. They're the same ones that a lot of the organizations use to confirm and label gluten-free food. And we use both of them because they capture slightly different parts of the gluten protein. So you could have a situation where one is positive and one is negative, and you would still have the potential to have celiac-related damage. So if they both look the same, you have greater confidence in the results. And again, I'm not going to go through each of these rows, but they're just different doses of the enzyme, 900 milligrams, 600 milligrams, or 300 milligrams, different doses of gluten, 3 grams, 1 gram, or 6 grams of gluten, so a fair amount in all cases, and with and without proton pump inhibitors to see if that made any difference. And again, what you see across the bold phase, which is the median degradation results in all these arms, is that you're really up to 99, around 99 in most cases, % gluten degradation across all these arms. So really sort of remarkable results, and I think worthy of further study. To just look at this in a slightly different way, this is one of those experiments where we actually looked at gluten degradation at 3 different time points: 20 minutes post-meal, 35 minutes post-meal, and 65 minutes post-meal. And versus %, this actually looks at milligrams of gluten digested, which can be a little bit easier to understand. So as we know, we typically talk about 50 milligrams as a safe threshold. Things below 50 milligrams would generally be considered to be tolerable by patients with celiac disease, and this is to some extent where the 20 parts per million comes from. And I just want to go straight to these circled boxes. So in the circle over here, you see on placebo, we put in about 1,000 milligrams of gluten and got out about 1,000 milligrams of gluten. A little bit less because you can't get all of it, but pretty close. With TAC-062, even at the lowest dose of 300 milligrams, you see the gluten levels the median is right around or below 10 milligrams, even by 20 minutes. So you're basically taking 1,000 milligrams of gluten, a slice of bread, and making it safe in this one experiment, at least in a smoothie form for patients with celiac disease. And this is well beyond what previous enzymes have been able to show. So otherwise, it was, as you would hope a phase 1 study would be, not too exciting. It's not absorbed. We didn't find any evidence of this in the blood, and there was no side effect profile that we were able to identify in this study. So to summarize the 062, it looks like from these data, both liquid and capsule formulations of 062 are really effective at degrading gluten in healthy adults. It was a favorable safety profile. And so we believe this is a really promising mode for potential treatment for celiac disease. And we are planning on launching a phase 2 study looking at the ability of TAC-062 to improve symptoms in small intestinal mucosal injury in patients with celiac disease. And we're hoping, COVID allowing, that we'll be able to begin that late this year. So we are actively planning for that and are excited to take this to the next stage. The next part of my talk was going to be TAC-101, but you already heard about that. So I am just going to focus on 2 main points, just to reiterate them, because this is a complicated therapy and a very novel type of therapy, much more unique than the glutenase, for instance. But really, this acts as, one way to think about this is this acts as a Trojan horse. You basically stick your protein, gluten in this case, in these nanoparticles that Ciaran talked about, and you use it to trick the body that these are harmless. So they turn from being something that the body reacts to being something that the body suddenly recognizes as not harmful. And it's all in the way these get taken up and in the way these interact with the immune system. And that's really the magic of this therapy, is the nanoparticles are very specially designed to be absorbed, to be taken up in special parts of the body that are where your immune system learns what is friend and what is foe. And so by introducing them in a place that they typically see friend antibody and antigens, friend proteins, it can turn off the immune reaction. And that's the data that you just saw, really the first ever data that suggests an antigen-specific immune tolerance can be achieved in people, in humans with an autoimmune disease. The one other thing I wanted to talk about quickly on this therapy, and because as Ciaran did cover this very, very well, is the symptoms. And I think one of the things that is often a little confusing in celiac disease is that we do our first clinical trials often, not always, but often with gluten challenge. And this means we take patients who are well controlled with celiac disease, give them varying amounts of gluten, and see if we can prevent people from having celiac disease immune activation. So that can be symptoms, that can be intestinal damage, that can be other biomarkers like the immune white blood cell T cell activation results that Ciaran talked about in the last talk. Some of these are pretty easy in their objective because the body either sees gluten or doesn't and reacts to it. Symptoms are really challenging though, and I would caution people look at symptom data from these gluten challenges and draw conclusions that I'm often a little bit of concerned about. So the two problems are this, that in general, the people who sign up for gluten challenge studies are the ones who have less symptoms when they get exposed to gluten. Because obviously, if you know you're going to be critically ill after eating gluten, you're not going to sign up for one of these studies. So first, the population isn't always exactly generalizable to the overall population. And second is the fact that people know they're getting gluten. So even knowing you're going to get gluten, even before you get it, most people will start to not feel well. And this is the same. This is not unique to celiac disease, right? If I was to tell somebody that you were about to eat a food that likely was, old and had, was contaminated in some other way with bacteria, people would start to feel sick whether or not it actually was. So that preconception of the fact that you're going to eat something that's going to make you sick makes symptoms really hard to interpret. So in this case, we didn't see any difference in symptoms between the people who were treated with TAC-101 and placebo, but that really wasn't one of the main aims of the study. Also, we gave a very big dose of gluten. So initially 12 grams. So about half a loaf of bread, not a slice of bread, but a whole loaf, to get the T cells to respond in a predictable way. So just to be cautious that the studies that we do in the beginning to really understand whether a celiac disease therapy might work, don't always apply directly to actually real life exposures, small gluten exposures. And so you could see therapies potentially like this one that didn't have much of a signal in symptom amelioration with very, very large gluten impact. But once we get them into the real world, they could make a big difference. So just to conclude for 101, and then no doubt there will be some further questions on this, these data were also positive enough to support further evaluation. We're actually planning a further phase 2 study coming up, hopefully starting this fall, maybe even as early as September, to understand more of how TAC-101 affects the immune system in celiac disease and how long the duration of effect lasts for this therapy. Some of the critical questions we will need to understand how to take this therapy forward and what it could potentially do to patients. So with that, I want to conclude with just a few general points. And I think the first one. There's no immune-mediated disorder that has a single accepted treatment. And we've had that for a while in celiac disease. For a very long time, we've had only the gluten-free diet as the only thing we could do. And there's no other immune disorder where that's the case. There's no immune disorder where that's even the concept that there would be a one-size-fits-all treatment. And I think we're finally getting to that stage in celiac disease where we understand that we're not going to have a single therapy for everyone, and that different patients are going to do best with different types of therapies that target different parts of the disease. And I really don't see this as we need to, that we'll have one drug that solves everyone's problems, just like the gluten-free diet doesn't solve everyone's problems. I think that, what these therapies will mean for patients is that almost everyone, at least for the near future, will stay on a gluten-free diet. But in some cases, these drugs will be used to help control the celiac disease when the celiac disease is not well controlled on a gluten-free diet alone. In other cases, the main benefit of therapy would actually be reducing the burden of celiac disease in the gluten-free diet, meaning that you maintain good control, but it doesn't command the same degree of constant vigilance, of burden, of lifestyle impact that it does today. And I think it'll be interesting to see as we get different therapies that hit different parts of the pathway, which ones work better for which type of people. So I think it's a really exciting time in celiac disease history. It's a very positive time where we have a huge amount of resources and intellect and enthusiasm for studying celiac disease and making lives better for people in the future. But the only way to move this forward, and I end almost every talk with this, is saying that we need people to participate, because on our side, we can put as much time and effort in as we want, but if people aren't going to show up to help with research, whether it's filling out a survey or taking part of an interventional trial or anything in between, we really can't move this field forward. And I really think this is an amazing time where you'll be able to be a part of history in changing the way we look at celiac disease for future generations. So thank you so much for listening, and happy to answer any questions at the end. And I'll pass it back to you, Vanessa. Thanks.
Thank you so much, Dr. Leffler. That was great. So now we are going to ask Dr. Leone to join us, and he will continue the discussion, and then we will get to everyone's questions, we promise. And the good news is that we are not, strapped for time having to run to the next thing. So if we run a couple of minutes over, we will do our best that we can to get everyone's questions answered, because I know there's a lot of them. So Dr. Leone, I will turn it to you.
Apologies. Well, thank you very much for the invitation. I hope next year we can do the presentations and the interaction live. Thank you to the DC Gluten-Free Expo organizers. Vanessa, thank you. I'm going to cover a bit more of the topic of drug development in celiac disease, perhaps a bit more high level than my colleagues, but with the same message, a message of optimism and hope. We are really coming of age in celiac disease. As you know, we all have to disclose where we're coming from. In addition to being the Chief Scientific Officer of Provention, I was formerly the CEO and CMO of Celiac and formerly CMO of Alba Therapeutics. I've been working in celiac disease for many years and in autoimmunity for many years. And the reason to focus on celiac disease is that really celiac is, despite affecting 3 million Americans, is the only autoimmune disease that has this kind of impact where there is no therapy approved. And there are many reasons for that. The gluten-free diet has been a blessing and a curse. The blessing is because it reduces the symptoms, and if you manage to do it properly, it will prevent you from having disease activity. The problem is the gluten-free diet has huge burden, and even for folks who manage to do the gluten-free diet correctly as much as they can, it's very hard to avoid gluten 100%. And as little as a few crumbs of gluten a day hidden in your meals will cause disease activity. Dr. Leffler and his collaborators published this important study showing that celiac disease treatment, the gluten-free diet, was the second most burdensome disease perceived by patients with many different diseases. It was felt as more burdensome than, for example, having diabetes or congenital congestive heart failure or inflammatory bowel disease. And it was only second to having end-stage renal disease, which is when you need to go to dialysis every couple of weeks or even more frequently. So a very burdensome situation. And that's why patients want solutions. Patients want either an adjunct to the gluten-free diet so that if they do it, it will work for them. And in the future, hopefully a replacement for the diet. That's not where we are just yet, but perhaps one day. Why it has taken so long? Well, drug development is one of the more difficult businesses, most difficult enterprises you can think of. It takes a very long time and a lot of money to develop medications. We ask investors to provide us with funds to test multiple drugs in animals, preclinical studies, eventually get into the clinics. And you need to have at least 5 to 10 drugs in early stage development to start to have some guarantee that you're going to make it through the finish line and get an approval. Marilyn Geller will tell you later the efforts to increase funding for celiac disease is a very important effort. The beauty of celiac disease for an immunologist like myself is, as Dr. Leffler described, we understand celiac relatively well. We all use this cartoon, and this cartoon just describes how gluten gets into the gut wall, activates the immune system, the T cells, and these cells called the IELs, the intraepithelial lymphocytes, which are the culprits of destroying the epithelial cells that are the lining of the gut wall, resulting in complications. A few years ago, in 2010, there were only 5 programs developing medicines for celiac disease. And just as an indication of how risky this business is, all of these companies that you see here no longer exist. I know many of you are especially disappointed by the recent failure of Immucent that had to cease operations when their phase 2 trial didn't show protection from a gluten challenge. It really takes a long time and a lot of research, and that's why we need you, the patient community, to help contribute to all of this clinical research. Fortunately, things have changed a lot, and as my colleagues indicated, we now have multiple approaches covering every step of the disease process. This will also help us with combinations. I'm not going to talk about all of this, just mention that we have approaches to prevent gluten from entering, and Dan talked about TAC-062 to digest gluten in the gut wall, drugs to prevent gluten from entering into the gut and from activating the immune system, the T cells, drugs to prevent the activation of the intraepithelial lymphocytes, and I will talk about anti-interleukin 15, Provention Bio's drug, and all the immune tolerance medications that Dr. Kelly described as well. So really exciting times in celiac disease. Provention Bio is the first company that has declared as our goal to try to intercept and prevent disease early, rather than waiting for the complications. So we want to tackle genetic predisposition and the early triggers of disease, and we want to tackle early inflammatory processes. Our most advanced drug has begun to show that this is actually feasible. Teplizumab, an anti-CD3 antibody, which is now being filed with the FDA for the prevention and delay of the onset of type 1 diabetes. After a single course of teplizumab, you achieve several years worth of clinical disease-free status. We believe if this can be done with type 1 diabetes, we'll be able to do it with celiac one day as well. We also have a vaccine, PRV-101, to prevent the infection, which has been shown to be associated with type 1 diabetes and celiac. It's called Coxsackie B virus infection. and we are entering into phase 1 clinical trials this year. And finally, I will spend a bit more time telling you about PRV-015, our interleukin-15 drug for celiac disease, which is partnered with Amgen. Interleukin-15 is the key stimulator of intraepithelial lymphocytes, and if you block it, you should be able to ameliorate celiac disease, reduce the activation of the immune system by gluten. We have a substantial safety profile already gathered in over 250 subjects. The drug worked quite well in phase 2 in rheumatoid arthritis, and then we took it into celiac disease. These are the results of the first phase 2 proof of concept study conducted in Finland with Markku Mäki's group. It was a gluten challenge, a very long challenge, 10 weeks of gluten challenge with a high dose equivalent to 1 slice of bread a day. You wonder, why do we torture patients? If you don't do a gluten challenge study, you don't know if a drug works, really. It's the only way to get an early read of the efficacy of a drug. This is precisely where Immuson T couldn't prove efficacy in the context of a gluten challenge. We had 3 doses of drug, low dose, high dose, against placebo. And what you can see here is the results of the trial. The gluten challenge period is in the box, and these are symptoms as measured by patient-reported outcomes. These are questionnaires that the regulators accept as valid indication of the efficacy of a drug. The placebo arm saw an increase in symptoms in response to gluten until the point when patients actually began to not take the gluten challenge. We were able to determine that this is what happened here because we measured gluten in stool and patients were not eating the gluten at the end of the study because of all the symptoms. So what you can see is that in blue and in green, there was a reduction in symptoms with PRV-015. If you look at other questionnaires like the celiac disease GSRS, there is also a reduction in symptoms significantly from a statistical point of view. The Bristol Stool Form Scale, which I'm sure is your favorite questionnaire, because you have to look at your stools and enter a number in your cell phone. It's a very fun activity, but it's really important to determine if a drug works or not, because what you can see is with placebo, in the placebo arm, there was an increase in diarrhea as measured by the BSFS. On drug, both doses of drug significantly reduced the impact of diarrhea, even below the baseline in the trial. And finally, the physician global assessment also showed that this drug was able to prevent activation of the disease. Physicians rated patients on the high dose of the drug as not having active disease. We also showed that there was prevention of inflammation or reduction in inflammation in the gut. This is the first drug that has shown both reduction in symptoms and inflammation. With regards to histology, the drug did not allow consumption of high amounts of gluten. We only showed the trend towards protection of the gut histology in response to smaller amounts of gluten. So this medication, while we believe it has a very good chance to be the first drug that will be approved for celiac disease, it's not going to allow patients to eat anything they want. What it will do is to protect them from contamination. It will be a monthly subcutaneous injection to be given at home, like an insulin shot. Once a month, it will protect you from contamination. That is our expectation. Obviously, we still need to conduct the next phase of the studies. There were no serious adverse events, and because of this well-tolerated safety profile, we have now been able to move to a much longer study, the proactive celiac study. It's a phase 2b trial. Phase 2b is the last stage between you go into confirmatory phase 3 trials. Proactive will be a 6 months trial where we will test in 220 subjects with celiac disease who are not responding well to the gluten-free diet. We will test 3 doses of drug versus placebo. Again, these are subjects who despite the diet, continue to have symptoms. There will be no gluten challenge here. This is now finally, we passed that stage. We are at the stage of real life, real world exposure to contamination with gluten. We will hear from Jocelyn Sylvester later how trials are conducted, so I'm not going to spend time here. But I will just mention there is a biopsy at the beginning and at the end of the study because it's important to show that the drug does not result in a worsening of histology and potentially shows in an improvement in histology. So I just want to conclude my presentation by thanking all of you for your heroic efforts at supporting and advancing celiac research. Together, we are going to develop solutions that will improve the quality of life with people with celiac disease. And one day, we will be able, no doubt, to intercept and prevent this disease. Thank you.
Thank you so much, Dr. Leon. That was really great. We're now going to bring in Dr. Jocelyn Sylvester, and we've been chatting on this side, and so I think she's going to answer some of these questions that have come up, but then as soon as she's done, we will open it up for all the panelists to be answering your questions.
It's an honor to be here, especially in a panel with such eminent speakers. And I'm hoping to talk a little more in general about celiac disease and clinical trials. Hopefully the title of my talk is not too depressing. I think really the key part is that you can help us get where we need to go. So like the other presenters, I'm pleased to have disclosures because that means that there's actually interest in treatments for celiac disease, which is why we saw that long list of treatments available. I'm going to talk a little bit about how you go from something you think might treat celiac disease in the laboratory to a drug that somebody can prescribe to patients. I'm going to talk a little bit more about what's involved if you want to participate in a clinical trial for celiac disease and how you can help. So the first step is always the idea. And once you have an idea, it needs to be transferred into a treatment. And so even before we get to starting the clinical trials phase, there's this really important preclinical phase, which could be looking in test tube experience, animal experiments, computer simulations. And once that has gotten to a point where there's confidence that this is something that's good for humans, then this is when the FDA gets in and the team can go to the Food and Drug Administration and file what's called an Investigational New Drug Application, or an IND. What this does is it gets permission to be able to label something as for research use only and give it to the first people. And so in considering whether or not that's allowed, what the FDA thinks about is these preclinical animal studies looking at what happens to the medication, how it affects the animals, is the manufacturing safe, and then also critiquing the proposal of how to show that this is safe in humans. So when we think about showing that a drug can go from the investigational phase to the new drug phase, which is when you now have something that a doctor can prescribe to a patient, there's really 3 steps. The first one is all about safety. The second effect is about efficacy. And then the third one, as Dr. Leon was just saying, is more about safety and efficacy and proof of concept. So it seems easy, but unfortunately it's not that easy. And this phase here is often in pharmaceutical development referred to as the valley of death. And the reason for this is it's hard to be successful because lots of things can go wrong. So if we have 50 medications that get an IND, they get the green light to go into humans. In step 1, we meet the lions, and there's about a 15% success rate. So maybe it's too toxic, maybe it can't be absorbed, maybe there's some off-target effects, maybe there's issues with manufacturing or maybe there's funding issues. These medications don't go past step 1. Then we have step 2, which is where it might be that you can give it to patients, it doesn't harm them, but it doesn't really seem to do what we want it to do. And that's really what we saw with the NextVax study. Once you show that it does what you want to do, then of these 2 of 50 medications you have left, the success rate's much better because a lot of what needs to be done has already been proven. And so of these 50 medications, we would hope that 1 would get to the finish line. So Dr. Leon showed us a slide that had lots of medications on it, and hopefully some of them, including some that you heard about from Dr. Kelly and Dr. Leffler, will make it across the finish line and into your hands. So what's
Sylvester, I'm so sorry to interrupt you, but we can't see your slides.
Oh.
There we go. Thank you.
So as I was saying, there's really 3 phases to clinical trial, starting out with this investigational new drug process. And then there's the risk of failure, which is high. And so if you start with 50 medications, so you go through phase 1, phase 2, phase 3, you're going to have medications drop out, but hopefully have one that's successful. And so in each of these phases, there's different goals and different things that are needed. So for safety, this is usually one of the faster phases, one of the cheaper phases, because you don't need as many people, because it's looking for very general safety signals to suggest that you shouldn't go on to phase 2, where you're going to have hundreds of patients instead of dozens of patients. Because you have more patients, this is more expensive. You're also looking to see if it works, which means it's more intensive. And once you pass phase 2, then you can go into phase 3, which is more patients, usually more well-defined patients, and because it's more, again, more money. So these numbers here are based on the average cost in the last decade for drugs that were successful. This does not include the cost of all those drugs that made it to phase 1 and failed, phase 2 and failed, phase 3 and failed. So when you hear figures about it costing billions of dollars to make a new drug, that's trying to account for all the drugs that failed. So when we're doing these tests and trying to do trials for a drug, what is the FDA looking for? And really, what they want is something that benefits how an individual feels, functions, or survives, and that those benefits outweigh the risks. So that's really, when we're designing trials, what we hope that the study is going to show. Also, one of the considerations is how does the new drug compare to existing therapies? And this includes medications as well as non-medications, such as dietary therapies. And so in celiac disease, we have some tools that are good for safety and some that are good for efficacy. And so when we think about safety, we look at blood tests, sometimes just looking, is there effect on the liver, is there effect on the kidney, in these early phases when we're looking very generally. Sometimes in the animal studies, perhaps there's a concern about the heart, so they might want to look specifically at the heart in more detail. We also have ways to look and assess, is it causing damage to the small intestine, either using video capsules or using endoscopies. When we think about efficacy, now, so it's a question, does it work? Really, because we're thinking about how a patient feels or functions, because fortunately, celiac disease is a condition where pretty much everybody survives. That means that patient-reported outcomes are really the most important thing. And after that, we can often support that with blood tests such as serology or immune activation tests. It may be, depending on the type of treatment, that we need to show efficacy through looking at the intestinal villi, but really often biopsy is going to be more of a safety issue than an efficacy issue. So one of the things about celiac disease, and one of the reasons why it's so challenging to get a drug for, is that it's really different than pretty much every other condition that already has a drug. And so some of the things that make it unique and challenging are that there's a wide variability in symptoms, and this includes people who are asymptomatic. So if somebody doesn't have symptoms, how do you demonstrate that you've made an improvement in how they feel, function, and survive? Also, there's an existing gluten-free diet therapy, which is perceived as being very safe and cost-effective with few adverse effects. Now, we know, and we're increasingly realizing, that a gluten-free diet is not necessarily gluten-free, and that's one of the challenges for doing these studies, is if you're comparing to a gluten-free diet, how do you know that somebody's really on a gluten-free diet? Safety is measured in different ways, and adverse effects are measured in different ways, and certainly there's changes to how one lives one's life on a gluten-free diet that may not necessarily be well captured in some of these patient-reported outcomes. And it's cost-effective perhaps from the payer perspective because they're not paying for a drug, but for the patient who's paying more for a loaf of gluten-free bread, it's not necessarily cost-effective. So this is really a difficult challenge in celiac disease that really distinguishes it from a lot of other diseases. The other thing that's important is that if the treatment is an alternative to a gluten-free diet, then it needs to be tested by eating gluten. And most people with celiac disease have been told since their diagnosis not to eat gluten. And this makes entering a trial for celiac disease different because in most trials, there's a hope of clinical improvement. There's a hope that you get the drug and this is going to make you feel better. In celiac disease, we want to show that if we give you the drug, we can give you gluten and not make you feel worse. And since in order to do this comparison, we need to give some people a placebo, which is a non-active form of the drug, we know that in conducting this trial, we're probably going to make some people feel worse. And that's a real challenge to getting people to participate and finding the right people. So I think in general, there's sort of two types of treatments for celiac disease. One is to make a gluten-free diet more gluten-free by trying to have some treatment for some of this cross-contamination. So this would be adding a drug to a gluten-free diet. And another is to allow what I'm going to call, if you'll let me, a normal diet, which is a gluten-containing diet. And so this is a drug that affects how these immune cells respond, perhaps, such as the TAC101, that is going to mean that somebody could potentially eat gluten. And so how you test these drugs is differently. If you're making a gluten-free diet gluten-free, you're going to need these patient-reported outcomes and questionnaires. You're going to actually have to take the drug or a placebo. There's probably going to be blood tests. There may be small intestinal biopsies. And because it's so hard to control this baseline level of gluten, you may be asked to eat small amounts of gluten. And you'll probably be asked to continue your strict gluten-free diet. Now, if we're talking about allowing a normal diet, there's questionnaires, there's taking the drug or placebo, there's blood tests, there's probably going to be a gluten-free biopsy because this is a really important safety outcome when you're adding gluten. And the reason you need the biopsy and to add gluten is to show that it's safe to eat gluten, which is why eating gluten is an important step. And so when we look at these studies, there's sort of a general design. So if you're thinking about volunteering for a trial, what are we going to ask you to do? And it's usually divided into 4 steps, and you saw the specific schematics for some of these studies in each of the previous talks. But at a very general level, the first thing you do is you hear about the trial, you call us, you come for a screening visit. We're going to give you more information, you're going to agree to participate, and we're going to make sure that you meet the inclusion and exclusion criteria. And so this will involve a lot of questions, possibly some blood work. And one thing that's going to be really important is to confirm the diagnosis of celiac disease. So if you've had a biopsy for celiac disease, I'd strongly encourage you to get a copy of your biopsy report from the pathologist that actually states what they saw when they looked at your tissue, because this is going to be key to helping us determine whether or not you qualify for our study. Sometimes there'll be an additional screening visit if there's criteria on how healthy your villi have to look. Next, there's often a run-in period because of this issue that a gluten-free diet is not necessarily 100% gluten-free 100% of the time, just to see what your symptoms are like and what your feeling and functioning is like on your baseline diet. This is going to include more questionnaires, possibly blood work. Next is the treatment period. And for most of these studies, you're going to be taking a drug or a placebo, so you will not know whether you're getting something that's active or something that's not. And it may include eating gluten either while you're taking the treatment or after you're taking the treatment. And again, we're really going to want to monitor how you're doing. And so there's going to be more questions, and likely more blood. Then there's usually a follow-up period to see how you do after the treatment. It might be that this is when we give you gluten, or it might be on your usual gluten-free diet. And there may be more tests in order to see how you're doing. And so in general, that's what's involved in a clinical trial. And I think the hardest part about considering being in a gluten-free trial is this whole question of, why should I eat gluten for science? Why can't we just give gluten to mice and then give a drug to me? And I think the most important thing is that if you want a drug that replaces a gluten-free diet, then it has to be tested on people who are with celiac disease who are eating a gluten-containing diet. Now, this is something we think about very carefully and is a very important part of clinical trial design. And you will have noticed that many of us indicated that we consult or are part of industry. And when thinking about clinical trials, often there's very intense discussions about how much gluten is needed, how should that gluten be delivered, should there be options to change or decrease the amount of gluten. And if you're invited to participate in the trial, not only have the investigators considered this, but the FDA has also considered this too. So there's a lot that goes into this. And we're also learning more about potential options. And I think the important thing to remember is that as in any research study for a clinical trial too, if it gets to the point where you become too sick, there's always the option potentially to down dose the gluten or to stop the gluten challenge part of the study. So this gets us back to the FDA approval process. And the question is, what can I do to help? How can you help? And I think the most important thing is to volunteer. Now, remember in phase 1, this is safety. Sometimes this is done in healthy people, so you can volunteer your friends and family who may eat gluten. And we also need all of these tools, which aren't necessarily well developed in celiac disease, and we don't necessarily understand celiac disease very well in terms of what's the average amount of symptoms that people have, what type of symptoms are people having. And so participating in focus groups, completing surveys, completing questionnaires, doing blood tests, even though you're not taking a drug, might actually help us develop the tools we need to be able to evaluate some of these drugs you've heard about. Of course, there may be an intestinal biopsy required, and if you're in a drug trial, there's going to be a medication or a placebo, and it's very likely you're going to be asked to eat some amount of gluten, especially when you're getting into these drug-related studies. And I think it's really important to acknowledge some of the really pioneers in clinical trials for celiac disease. And these are the patients who eat gluten so that we can monitor their response and learn about the normal response to gluten before we're even able to study a drug. And so these are very altruistic people who are volunteering to eat gluten, knowing that there's no chance that they'll get a drug that might help them. And we really can't do it without these volunteers, and it's important to thank these people. So where are we going next? Although there's lots of things that could go wrong, I'm optimistic. I hope it's going to be an all-you-can-eat buffet. And how we're going to get there is with time, money, and most importantly, you, because we can only get a treatment for celiac disease if people with celiac disease help us test the treatment. And so put yourself in the picture. This is some of our patients in our cohort study in Canada, and we need more volunteers like them. And again, thank you to Vanessa and Children's National for organizing, and I'll stop now so that we have time for questions.
Thank you so much, Dr. Sylvester. And Sandy and I would love it if all of our panelists could rejoin us. So there have been quite a few studies from people in our audience asking how they can get involved with these studies. Do they ask their doctors? Do they contact you guys? What should they do? And can both adults and children be involved?
All right, if nobody wants to jump in, I'll jump in. So there's different levels of study. So when you're talking about clinical trials, once you have that investigational new drug and you're testing a drug for research, these studies you could find by looking at clinicaltrials.gov, which is an index of all the trials that are currently going on around the world. So it may not be a study that's near you, it may not be a study in the United States, but it's a good way to learn what's happening. Be connected to your celiac center and to your physicians. Ask questions, join your celiac organizations, join your support groups, because through events like this, you're going to learn about things. And read reports, read the news, follow what's happening.
Great. Thank you so much, Dr. Sylvester. That was a wonderful presentation, and I hope you will get many people interested and dedicated to clinical trials as a result of your presentation today. So it's absolutely critical for the future of everyone with celiac disease. So join in, everyone. And here we go with some questions. Here's one from Vanessa's mom. She asked, did people have symptoms from taking a gluten challenge? And if so, I'll add to that, what kind of symptoms did they have, and what did you notice about them?
I can address that since I showed you some data from the gluten challenge study. Yes, people frequently get symptoms related to a gluten challenge. As Dan mentioned, we tend to discourage people who get very severe gluten-related symptoms from participating in a gluten challenge study, because we don't want people to become very ill and have to stop the study because they're feeling terrible. And so we generally we have a selected group in the study who have less severe symptoms. So symptoms that would be typical would be nausea, abdominal discomfort, loose bowel movements. But typically the symptoms resolve once the gluten challenge ends. The gluten challenge studies vary. Sometimes the gluten challenge is as short as 3 days. Sometimes it's as long as 12 weeks. So generally, the symptoms are most severe at the beginning of the gluten challenge, and they tend to become less severe as the gluten challenge evolves. But as Dan said, we really discourage people who have terrible symptoms from participating, and we prefer people who experience mild, tolerable symptoms.
Mm-hmm. And that brings up a point that maybe you would like to address, which is, are you seeing, and really this could be addressed to any of the panelists, are you seeing as you go along in your developing your expertise and information and knowledge in celiac disease, are you seeing a wide range of intolerance of gluten and symptoms with regard to mildness ranging to severity?
I'm not sure there with your question if you're asking about celiac disease or non-celiac.
Oh, celiac disease. Well, actually, I mean, that's a good point, because, and how is, and maybe you want to tell our listeners how the non-celiac gluten sensitivity actually relates to celiac disease. Maybe just say a couple of words about that.
Well, they're separate conditions, clearly, as the name non-celiac indicates. These are individuals who have symptoms related to celiac ingestion who do not have celiac disease, which is an autoimmune inflammatory disorder. People with non-celiac do not show the same inflammatory damage in their intestine as those with celiac disease. And I think all of the presentations today have related to celiac disease and not yet to non-celiac gluten sensitivity. But within the realm of celiac disease, absolutely yes, is the answer to your question. Some people with celiac disease have terrible symptoms, severe diarrhea, weight loss, and malabsorption. And others, in fact, many with celiac disease have no symptoms whatsoever. So there's a huge spectrum of symptom response.
Mm-hmm.
So I'm going to try and combine a number of questions in one. There are a lot of questions about, do you have to have one celiac gene or the other in order to take these drugs? And would you not be able to take them if you had a coexisting autoimmune condition, like a thyroid disease or Down syndrome or diabetes?
Yeah, I can try to address this. So those are great questions about inclusion and what you need to know. And as Jocelyn mentioned, the most important thing you need to have is proof that you were actually diagnosed with celiac disease by a clinician. And in most cases, that's going to be a biopsy, although there are some trials that may have some wiggle room on that. But that is an issue, right? Because we know that sometimes the diagnosis is made based on lesser criteria. And that's one of the reasons that most studies will test for the HLA DQ2 or DQ8 at the study entry, just as another confirmation that you actually have celiac disease. And because there's a high probability, although it's not 100%, that if people who don't carry those genes will not have celiac disease, most studies will exclude those participants. But on the other hand, getting that information, if you don't already know it, is actually one of the potential benefits of the study, is you'll get your HLA information, as well as celiac serologies and some other things. So there is some other information. The second part of the question was on, what was the second part of the question, Vanessa?
If you have a coexisting
coexisting disorders, yeah.
Can you still be participating?
Yeah, so that's another thing that is just very study specific. In general, because things like thyroid disease are so common, as long as that's your thyroid disease is well controlled, that usually will not preclude you from study entry. That may also be the same for some studies for type 1 diabetes if it's well controlled. If you start getting into other disorders that are more complicated, where you're on immunosuppressants, it's going to exclude you from most studies.
Great.
Good. Katie Lamerson asks, with regeneration of blood cells, how often would you expect to have to take the treatment with that particular
So this is, I assume, the TAC 101. Typically, yeah. Ciaran, do you want to try to address that?
Well, the answer is that neither Dan nor I know the answer, basically. And that's one of the questions that's going to begin to be addressed by the further studies. The studies that I presented were very short-term studies. And so whether tolerance lasts a week or a month or a year or a decade, we don't know. And obviously that's one of the big questions that needs to be addressed in the further studies that Dan is planning.
She also asks, Katie Lemerson continues and asks, also, there was much reduced ELISpot activity and reduced villi atrophy, but it wasn't zero. Would there still be symptoms with this reduction?
Well, as Dan alluded to, symptoms are an essential measure of the efficacy or effectiveness of a drug, but symptoms are somewhat are a difficult measure to use because symptoms are subjective, and so people will think they're getting gluten, and so they'll feel they're sick, or they're going to be robust and think I'm not going to be sick, etc. So they're very subjective. So that was a very small study in a small number of patients, and so symptoms were not a major outcome measure for that study, but they will have to be examined more closely in the future studies.
So we've had a couple of questions about wanting to better understand the difference between developing a vaccine for COVID-19 versus a vaccine for celiac disease. Why do we think it's possible to develop a vaccine for COVID so quickly, but it takes so long for celiac disease?
Well, you know, a celiac vaccine is really an anti-vaccine. Usually when you're developing a vaccine, you're trying to train the immune system to react against and attack an opponent, be it the flu or COVID or rubella or whatever you're trying to activate a response. With celiac disease, you're trying to do the exact opposite. You're trying to deactivate a response. You're trying to not vaccinate, but tolerize the individual. So this hasn't been done before. And so I don't think anyone can predict how long it's going to take. But I do know that Dan and his colleagues and others will be working really hard towards the goal. It's a really interesting task that is being taken here. Dan, do you want to comment further?
Actually, I would be interested. Yeah, I mean, I agree with all that. I'd be interested to see what Francisco, because he's developing a prophylactic vaccine, and how he thinks that fits in, because that might be more similar in this case to the COVID vaccine.
Francisco, he can put his video up. I think he can.
I can see him. Yeah, I can see you.
Oh, okay.
Yeah. So that Dan and Ciaran are correct. There are two types of vaccines. One is the prophylactic vaccine. It's also called primary prevention. You prevent an infectious trigger of the autoimmune disease or the infection in your body. In our case, Provention, we are developing a vaccine for Coxsackie B virus because this virus has been linked to celiac, a subset of celiac, and a subset of T1D, type 1 diabetes. Even that is not fast to develop these kind of vaccines, but for COVID, obviously the entire medical community is behind the effort. Billions of dollars are being applied, and that's why it's being accelerated. The second type of vaccine is the one Ciaran described, and that is a secondary vaccine to desensitize the immune system against attacking ourselves. And that is even harder. It hasn't been done yet. And as Ciaran said, probably the drug that Takeda is developing with Dr. Kelly and Dr. Leffler, it's probably going to be the first. We hope it will work. So, I think if I summarize and answer some of these questions that came earlier, there are like three different types of drugs. First, you have drugs that you'll take every day with your meal. That's like the glutenase that Dan is developing. You take it with every meal, and it will digest gluten, reduce the amount of gluten you have entering your body. The second type of drug will be the one I am developing. You block the immune response against gluten at the level of the innate immunity. This is the first defense in the gut, at the level of the gut lining. We think we can give that vaccine once that drug once a month to prevent the effect of gluten contamination. And then finally, they are trying to act deeper at the level of immune tolerance. If they succeed at developing a drug for immune tolerance, it potentially could be much less frequent. It could be once a year, once every 2 years, who knows? And I mentioned earlier that at Provention, we have such a drug now for type 1 diabetes, where a single course provided years of being free from clinical type 1 diabetes. So it's theoretically possible to do the same in celiac. We just have to keep at it and keep doing clinical trials.
So we are getting lots and lots of questions, both email, text message, in the box from parents of high school and college students who are wondering how their children who have celiac disease and are really interested in all of this work can be involved. First, can they join the studies, and do any of your organizations have internships that they could apply for to be more involved?
I can start. Three answers to that. First, and this is with the National Children's National Hospital. We are planning some basic research work with Dr. Kersner. We're going to study patient-reported outcomes in children and adolescents. That will be one opportunity to participate. The trials, they usually start in adults first, because that's by regulation. You have to show some safety and efficacy in adults first. In the case of the trial, Provention is going to start in August, our phase 2b trial that doesn't require a gluten challenge. We are going down to 18 years of age, so we can take some older adolescents, but not younger kids. And then finally, the question about internships. I think bigger companies like Takeda may have great programs for this. Provention is a small company, so we don't have that kind of program at this time, but contact Vanessa and we see what we can do.
Here's an interesting question. Lisa Harding asks, in TAK or TAC 101, does the nanoparticle dissolve in the liver, spleen, and such, or does the gluten stay in the pocket until it is removed from the body? Does it have to dissolve for the body to have the opportunity to see gluten as a substance to tolerate? That'd be great to clarify.
Sure. Yeah. Yes, indeed. The nanoparticle is very quickly filtered by the spleen and the liver as it goes through the circulation. And once it's filtered in those organs, it's trapped and very quickly degrades. So as I mentioned, the substance is the same as in biodegradable stitches or sutures. So it's then dissolved and then it releases the gluten. And that's the key step, because if it's not released so that it can be accessed by those Marco cells that I mentioned, then it can't work. So yes, it circulates, it's trapped, it's released, and then it has its effect.
Well, here's a good one for Dr. Sylvester. Her study, doggy bag study, noted that people weren't great at knowing when they were actually glutened. Does this affect drug studies if people aren't good at knowing when they are actually having symptoms from gluten?
Yes, this is a tremendous problem, and one of the biggest challenges in making a drug that's an adjunct, something to take in addition to a gluten-free diet, because it's really hard to know what the gluten-free diet is, and it probably varies between people and even within an individual. The more we study gluten exposure, the more it appears to be something that's somewhat random and episodic. And so this is part of the reason why, if somebody's studying a drug to look at a treatment for gluten exposure, part of the study may involve having low doses of gluten, such as the amounts we are finding in our study, where people may not necessarily have had symptoms, but they did get a gluten exposure. And so it's something that can be accommodated in study design, but it's definitely a problem and definitely also a challenge in that it might make the gluten-free diet seem better than it is, because people can't report when they have gluten, so they presume that they're gluten-free.
And to just piggyback on that, why do you think more people in the placebo group reported symptoms?
Yeah, I can answer that. That's by design. We give a gluten challenge to trigger symptoms and then to try to measure the efficacy of the drug comparing with the symptoms in the placebo arm. If you recall, we saw a nice increase in symptoms progressively until the point when patients stopped taking the gluten challenge, and there was a reduction across the board with the drug, with the anti-interleukin 15 antibody. That is exactly what you want to see. Now, the next trial will not have a gluten challenge, so then we will have the issue that Dr. Sylvester just described, which is we will not know exactly how much gluten people will be exposed to in their real world diet, but we will measure that by collecting stool and urine and looking at the gluten content to try to understand who has been glutened and did the drug protect them or not.
Here's a last question to wrap up. There are a lot more, and we will certainly address those questions later in writing, so you guys can keep asking your questions. But N. Spain asks, are children diagnosed with celiac disease considered at risk for type 1 diabetes? And are they then eligible for the vaccine that Dr. Leon talked about in his presentation? And she also adds, just before you answer that question, Dr. Leon, thank you for your presentations and all the time and effort you've put forth for type 1 diabetes research as well as celiac disease.
Oh, thank you for that kind comment. Type 1 diabetes and celiac are so similar in so many ways. They share the genetic predisposition, they share some of the viral triggers, and they are definitely linked. We need to learn from each other. We need to look at what type 1 diabetes is doing better than we're doing in celiac and vice versa. So it's important there is crosstalk. The only thing I want to clarify about the vaccine is that this vaccine will only work in very young kids, in neonates at birth, because Coxsackie B virus is present all throughout the world. And if you don't get the vaccine in the first 6 months of life, you will not be able to prevent the infection. So we are targeting the vaccine as a universal vaccination to be given in the first few months of life. And hopefully we will be able to reduce both type 1 diabetes and celiac substantially. It's not going to be perfect. There are other viruses involved. But if we can reduce the diseases by 10, 20, 30%, that will be phenomenal.
Well, I want to and thank all of our speakers. This was an absolutely amazing session. We know there are still questions out there, and we will make sure to get them all answered and posted when we put up all of the video recordings. It's just been so great to hear about all of these developments and research, and what a bright future we have for our celiac community. So it's now time for another break for everyone to grab some lunch, but please don't forget to come back at 1 o'clock for the next session. That session will be with the Celiac Disease Foundation, where we're going to talk about advocating to advance celiac research on Capitol Hill, as well as at school. So we will see you all back here shortly.
Thank you very much.
Thank you all.
Thank you, everyone.
Thank you.
Thank you.
Yeah.